Hey everyone, good morning. This is JD Harwell, and I'm going to be presenting along with my facilitator today, Austin Pepperling. And we're going to be talking about a new paper that was just published last week in the Journal of Neurochemistry. This is exciting research, answers some questions that have been there since 1965 or so. The paper's titled Heterogeneous Glutamatergic Receptor mRNA Expression Across Phrenic Motor Neurons in Rats. Research was done by Sabia Reina, Gary Seek, and Dr. Carlos Mantilla at the Mayo Clinic in Rochester, Minnesota. Now, before we go any further, I just want to do a little thought exercise. I want you to think about your breathing right now. Think about your breathing. Of course, we have our lungs. I'm going to do my best to just not butcher a set of lungs here. So we've got our lungs, and you're probably breathing, fairly slowly right now. Other times when you're exercising, or maybe you're huffing it across lower campus up to Westridge, you start breathing faster and faster. And I've never really thought about this. I mean, I know that we have our diaphragm down here. So we have our diaphragm. We have our diaphragm that's facilitating this inspiration. But I never really considered that the diaphragm is not homogeneous across. If you think about weightlifting or cross-country running or sports, you know you have fast twitch fibers, slow twitch fibers. And in fact, as far as the diaphragm goes, you have these different motor units that are comprised of slow twitch fibers, fast twitch intermediate, fast twitch fatigable, and they have to be able to control our breathing at different rates. So the question then arises, how are these things, how are the individual motor units innervated, I guess, and I guess we know they're innervated by the phrenic nerve, but the question is, how do we activate certain ones at certain times? And this paper helps to answer that, or at least gives us another component into the question. Now, I used this image of a neuron on the left that I've got the laser pointer on here. I used this as this background because I want us to think about this picture of a neuron instead of this one that comes to mind. So as soon as someone talks about a neuron, I always think of this picture from the textbook, this cutaway two-dimensional, and I want us not to think about that because we're focusing in this paper entirely on the soma. So if we look at this, if I think of the surface area of this, I'm really thinking of the surface area of this two-dimensional thing. But what the paper is talking about is the actual surface area of this spheroid somal body over here on the left. So this is what I want you to think about. This is the region we care about. We don't care about the axons, the dendrites in this case. And in fact, for this paper, they only look at, in this research, we're only looking at phrenic motor neurons that have a visible nucleolus. So we have a very visible nucleolus, and that's important because when the nucleolus is very visible, we know that we must be creating ribosomal RNA. And if we're creating ribosomal RNA somewhere out in the cytosol, we're going to probably have mRNA transcripts. Okay, so there are a few abbreviations that we need to pay attention to when reading this paper or talking about the research. The first is AMPA. You can see what it stands for right here. The second is NMDA. These are the two most important ones. Now, you don't need to worry about this, and I didn't need to worry about this, because what we really just want to say is glutamatergic receptors 1 and 2. In the paper, they'll talk about they use the terms GRIA2 and GRIN1 almost interchangeably with these. But what this is the AMPA and the NMDA. These are the actual receptors. And the GRIA2 and GRIN1, these are the genes associated with those. So the paper sometimes uses one, sometimes uses the other. It's important to know what they're talking about, and I probably didn't figure that out until well after I had finished the paper. When you see PHMN, either in the paper or in this presentation, it's referring to the Phrenic motor neuron. And the Phrenic motor neuron, it's important to also recall that that is the nerve. The phrenic nerve is the one that innervates the diaphragm. So it's not innervated by anything else, as far as I know, at least from AMP. Okay, CTB 488. This one's important because as we talk about labeling the neurons, this is the chemical that they did it with. This is right here. It's a cholera toxin subunit, and that's what's going to be used to fluoresce later. And then this is just the website, the RRID. This is where you can find more information about any of the research resources. Okay, so I already covered that. So what's the significance of this research? The first is that what I talked about before is the order of recruitment of these motor units within the neurons, or the neurons and the muscle itself. So when you're breathing lightly, you have the slow twitch that are going to be innervated. These are very, very resistant to fatigue. And we've always known that there's some sort of order. I can't say always, but at least since the 50s or 60s, they've known that these things fire in a particular order. And a scientist named Henneman came up with what's called the size principle back in 1965. And the size principle basically explained that the order had to do with the soma of the neuron. They weren't sure exactly why. What they thought was that a larger soma has more receptors, and so that would make sense that more receptors can bind more ligand, and then maybe that's how that happens. And in this paper, the scientists wanted to ask the question about whether receptor density on the neurons would matter, and whether or not it would support the size principle. So they had a hypothesis that AMPA and NMDA, and remember we can just always think of this as glutamatergic receptors. And glutamate is a fast-acting neurotransmitter. It's the one known, the known ligand for these. But the question is whether or not, or the hypothesis is that the receptor's mRNA expression would vary across the phrenic motor neurons, such that smaller phrenic motor neurons would have a higher density of mRNA transcripts compared to the larger ones. Now, how to think about this? So if we're just looking at the size principle, all right, so imagine all these golf balls, imagine all these being the soma of a motor neuron. So if you had larger surface area, you can have more. But what if you had the same number of divots? What if you had the same number of divots on the large ball as you did the small ball? If you did that, the density on this small ball would be much higher, right? And the thought is that this really could matter, because if you're able to bind more ligands here, because they're in such close proximity, you could alter the cell membrane potential much more dramatically than you could with these spaced out with these spaced out divots or receptors. Now, keep in mind that this large ball actually has more receptors overall, but if our hypothesis is correct, that would have a lower receptor density. And if you think about the cell membrane potential, it would make sense that you could fire this one much faster because you're able to alter the overall membrane potential more dramatically faster. Okay, so what was studied here? We have our rat. These are, what are they? The Sprague dolly rats. These are the guys that get recruited to do a lot of science. And in fact, they're going to do some science for us in this paper. So we have our Sprague dolly rat. And what they did was label the phrenic motor neurons using that CTB488 that we talked about. And they injected it between the 7th and 8th ribs into the pleural space. Now, I don't know why they did that. I don't quite understand it, I guess. When I think of the pleural space in the lungs, I'm thinking about the small space between the visceral and the parietal pleura. And I'm not sure why injection there would get to the motor neuron, but I guess maybe there's some retrograde transport. That's all I can think of, especially since they did this T-3 days prior to euthanizing the animals. Once they euthanized the animals, they excised regions C2 to C7, and then they put that on. They sliced it up, put it at minus 80, and held on to it until the research, until they could get to each one. So, we already mentioned that they only used phrenic motor neurons with a visible nucleolus. Here's what I was talking about. So our diaphragm is down here, right? And the injections went here. So I'm guessing that the way this worked is this CTB488 made it in here, and then when the and then somehow traveled maybe retrograde into the motor neurons. Once they harvested the motor neurons, they used what was called an RNA scope assay, and they were able to detect the receptor transcripts in situ. And the way that this works, the RNA scope works is much like most of assays that we do, but they put the cells and the tissue onto a plate. They hybridize the RNA that they're looking for. So this is the mRNA for the two receptors. And then they amplify that just like a PCR or something. But then they're able to actually see these with a microscope. Now the key for why this is good, why this particular assay is used, is because it allows for a very, very high signal to noise ratio. And that's because they're able to amplify the signal while suppressing the background simultaneously. And it's so effective that you're able to actually look at single molecules within single cells. And it works with basically anything you want to look at. So this is a very, very helpful assay. There were two controls. First, the positive control. They had a sample from the dentate gyrase region and the cerebellum. They did this because these are known regions where the two transcripts would be found in abundance. So that would be our positive assay. Negative assay was the negative control is they did the assay in the presence of RNAs, and by doing so, they know that everything would be degraded and they shouldn't see anything, and that was the case for both the positive and negative controls. Both worked well. What they wound up finding was exactly what they hypothesized, is that the density of both of the transcripts has a significant negative correlation with somal surface area. So as surface area went up, the mRNA transcript density went down. Now, they made a big assumption here. So they made this assumption. And the assumption was that mRNA expression correlates directly with, mRNA density rather, correlates directly with the protein concentration. So that is generally the case. I don't like them. I really don't like the paper being published going off that assumption, even though it is normally the case. It's not always the case, as I've learned in other classes. So I wish they would have actually looked at the proteins, but I guess the mRNA transcript is a lot easier to look at. But what they did find, the takeaway here, is that as the somal size increases, the density of the receptors decreases. Okay, so there are a number of figures, 8 figures that we want to talk about. And this first one is staining of different regions of the cell to see where the receptors are. Okay, so in this case, you can see nuclear regions. You can see these outer regions, and you can see where everything actually is. This is super interesting. So here's your different transcripts that we're concerned with and where they're located, whether they're in the nucleus, the cytosol. You can see the DAPI, DAPI. This is one that they also looked at here, but you can see it's not in the cytosol. It's only resides in the nucleus here. The NMDA is all about, and so is the AMPA. So they were able to see the stuff they were looking for, where they were looking for it. Now the question is quantifying it. All right, so they divided the somal cells into categories. They took the surface area, and rather than treating it continuously, they treated it as a discrete variable, divided it into tertiles, and looked at lower, middle, and upper. And you can ignore the triangles and circles. Let's totally ignore that and treat them all as dots or whatever. And the reason is that they did compare male to female expression, but they found no difference between sexes of the animals. What they did find, however, was that in the lower tertile, you had a higher density of, you've had a higher density of the mRNA transcripts. And then as you move into the top tertile, you can see that there is a lower density. And the colors represent individual animals here. And so you can pretty much ignore the color and ignore the shape and just look at the total number of measurements. They also, you can see like these outliers here. They did statistical analysis for outliers. They did find outliers, but they did not exclude anything. Every data point that they found is included in the figures. All right, figure 2, figure 3. So on figure 3, what we see is, if we look at each of these, imagine these both telling us the same story. This top one and the bottom one. And the reason is that one is AMPA and one is NMDA. And we're treating those the same only because they behave the same and they're both receptors for glutamate. But what you can see is that as the surface area increases, the total number of transcripts also increases. This is important because this goes back to the original thought that larger size soma would have more receptors. And that is true. We do find that. You can see the same thing when they take it off of a continuous spectrum and put it here as discrete. I think I said dichotomous earlier, but that's the wrong word. But when they treat these as discrete categories, you can see that the upper tertile does definitely have more receptors. All right, now we're not talking about absolute anymore. Now we're talking about density. And this is where the magic happens, I guess. And this is that as the surface area increases, here, the density actually goes down. So the density goes down. This is the big takeaway, because what we see then is that the lower the surface area, so if we look way down here, the lower the surface area, the higher the density of receptors. And so if the density of receptors is higher, we can change the membrane potential more easily. All right, figure 5. Figure 5 is interesting because what this does is show us this side is going to be nuclear and this side is cytosolic. So you can see that the nuclear transcripts do not change. They noted this is important, and but within the cytosol is where you see that negative correlation. All right, so they put all the data together, and this shows the density of AMPA versus the density of NMDA, and then they've divided it into the green regions. The green region is that upper tertile with the largest somal surface areas. The blue is the smallest, and the red then is in the middle. But this very clearly shows that as the somal size decreases, the density increases for both of these receptors. Okay, figure 7. Figure 7 they didn't use. And then, of course, this is figure 8. So we'll end with that. And let you know that if you have any questions on this paper, Austin would be happy to answer them. He can be reached at ajpepperling@alaska.edu. Thanks so much, everybody. Have a good day.